Good afternoon, and welcome to today's Congressional Neuroscience Caucus briefing, Advancing Neuroscience for Americans, Breakthroughs in Addiction Research Enabled by the NIH BRAIN Initiative. I'm Mark Resnick, Distinguished Professor of Physiology and Biophysics and Psychiatry at the University of Illinois College of Medicine, a career research scientist in the Veterans Administration, and most importantly, board chair of the American Brain Coalition, and I speak only for the American Brain Coalition today. So the American Brain Coalition, in partnership with the American Academy of Neurology, American College of Neuropsychopharmacology, Dana Foundation, IEEE Brain, Simons Foundation, Society for Neuroscience, and the Kavli Foundation, is proud to co-host today's briefing alongside the Congressional Neuroscience Caucus. And this morning, ABC leadership and representatives from several of our partner organizations have been meeting with members of Congress and their staff on the Hill. We've been highlighting the vital importance of continued federal investment in the NIH BRAIN Initiative, which is transforming our understanding of the brain and paving the way for breakthroughs in treating neurological and psychiatric conditions, including addiction. Could anyone not hear me?
Start over.
Start over. Nah. You want me to do that, right? Yeah. All right. So ABC is thrilled to work closely with the Congressional Neuroscience Caucus, co-chaired by Congressman Morgan Luttrell and Mike Thompson. And the Congressional Neuroscience Caucus was formed in 2010 to raise awareness of the essential role brain research plays in human health and society, highlight the progress and promise of neuroscience research, and promote federal policy that supports continued innovation in the field and both fairness and equality for those who suffer from brain diseases and conditions. Congressman Luttrell represents Texas's 8th district and is currently serving his, well, you're in your second term. Yeah, his second term. Congressman Luttrell is a former Navy SEAL who survived a 2009 helicopter crash that resulted in a traumatic brain injury, an experience that fuels his commitment to neuroscience and recovery research. After his military service, he earned a graduate degree in applied cognition and neuroscience from the University of Texas at Dallas. In addition to his role as congressman, he now works to support fellow veterans navigating PTSD and traumatic brain injury, and it's our privilege to welcome Congressman Luttrell.
Thank you so much. I was going to tell you them. We were over there, Mr. Thompson over there making jokes at you because the microphone went on, but we figured somebody would come up here and help you out. Good afternoon. It's always such an honor to come in and talk brain science because that's kind of one of those uncharted frontiers that nobody really likes to get in discussions with because the brain is this anomaly that sits between our ears that we are still trying to understand. I want to thank Mr. Thompson for jumping on board and allowing me to work alongside him in the Neuroscience Caucus. And as he and I navigate these challenging waters in Washington, D.C., I want to stand up in front of you all and say out loud that I personally recognize the importance of funding the NIH initiative, brain science, neuroscience initiatives when we're concentrating on research-based protocols for the brain and the human body. I think if the American public understood it, if we actually trained our brains and conducted research on our brains as we do our bodies, we would be we would be absolutely superhuman. The ability for us to one day tell a mother or a father that has an injured baby, that has an injured brain, say, hey, don't worry, we've got a hold of this. And unfortunately, we're just not there yet. Now, I know brain mapping and our understanding of neural connectivity and how neuroplasticity and changes and what that's shaping up to be. I've visited this fine young gentleman and some of the other doctors. I've got throwing you a shout out. Come on now. And just deep brain, just going into kind of deep brain research and our ability to touch areas of the brain that we didn't used to be able to do, and the changes that's manifesting in the population is amazing. Now, it is my job as a congressional member to make sure that we are eyes wide open and listening, because every time I have the opportunity to say I want to say we're there. I do. When I was going through school, my professors like, every single textbook that I had in grad school is no good anymore. We've learned so much. And now I'm saying that about my textbooks. It's like, hey, we've learned so much since that. And I've only been out of school for, almost a decade. But where are we? Are we touching the next phase of evolution when it comes to the understanding of how the brain works? Will we one day understand the mechanism? And then where will that lead us? And I hate to do this because I put the onus on the subject matter experts that are sitting at this table and those babies that are coming up through academia right now that are hungry, because we have to have that appetite. And unfortunately, it is such a small fractionated group of individuals that like to, I say necessarily dork out on the brain. You have to be able to do that. It's a very specific language that you have to understand. It's beneficial for me to be here because I can share that information with the members of Congress and the Senate and the administration and say, this is why this is necessary. When I was in grad school, I told my professors, I was like, I don't have any idea what you're saying. Can you put it in crayon for me? And I'm not saying I have to do that here, but there is an understanding of how you have to have those discussions. So I would say absolutely hang on every single word that everybody at this table is about to lay out for all of us, including those on the camera that are watching from afar. And it's one of these necessary, evils or necessities that you have to love. You have to understand that if you don't fight for this, if you don't have the ability to understand that we are going to lose again, lose some more, to gain enough information to when we have a win, and then we're unstoppable. So thank you for having me. Thank you for hosting me. I'm sure Mr. Thompson is going to come up next, but I'm sure there's going to be an amazing introduction for him, is that going to be the case?
Yes, and the microphone will be on.
And the microphone will be on. Okay, so thank you all. Have a very best day. God bless.
Dork out on neuroscience. I guess that sounds fine to me. That sounds fine to me. Congressman Mike Thompson represents California's 4th District. He joined as co-chair of the Congressional Neuroscience Caucus following the retirement of Representative Earl Blumenauer. Congressman Thompson brings a long-standing commitment to healthcare, mental health access, and scientific advancements, principles closely align with the goals of the caucus and today's discussion. Congressman Thompson is also a Vietnam veteran. We thank him for stepping into this leadership role and continuing the vital work of the Congressional Neuroscience Caucus.
Matt, thank you very much. I'm honored to be with you today, and I can't tell you how pleased I am that Earl Blumenauer asked me to step in and take his position as co-chair when he left. It's a real honor. And I didn't really know how I was going to like it, what we were going to be able to accomplish. I know clearly that there's a lot of work we do need to do. And Morgan and I hadn't known each other before. I think that lunch is the first time we met. I didn't know how it was going to work between he and I. And on the way over here, I didn't know how it was going to work when we're going to be sharing the podium at a time when the work that is so critical to our country and so critical to everything that all of us believe in hinges in the balance right now, given the issue of funding for some of the most critical research that we have to do. But when you talked about the funding, I thought, we can be together on this. I'm a big believer in the old saying that anybody who says you can't solve a problem by throwing money at it is talking about somebody else's problem. And we all know that. The thing that got me really interested and motivated in regard to mental health issues is it's glaringly obvious that we do not have the resources nor the data and research to address the work that needs to be done on the brain. We have spent an ungodly amount of money necessarily on patching, on putting Band-Aids on symptoms. And quite honestly, that's the most of the work that we've done in my career in public service, is dealing with symptoms. We don't have enough money to do it. We'll never have enough money to do it. And I'm a big believer that we need to invest on the other end to best understand the brain, best understand how to stop these symptoms from happening. And that's why I agreed to co-chair this caucus. That's why I'm excited about working with Morgan, working with all of you, working with Mike Kelly. He and I have a big bill that invests a lot of money through tax policy in brain research, something that can really be a game changer. So the work that you do is important. I'm all for the funding that's necessary to make sure that you're able to do your work, that we're able to retain the bright scientific minds that we have now, attract others, and don't create a barrier for people to get into this line of work, because it's critical. It's critical to all of us. There's not one person in this room who isn't this far away from mental illness, one way or another. And we need to make sure that we provide the best research and the best tools to be able to deal with it. So thanks very much. Thanks for letting me be part of it. And let's get this stuff done. Thanks.
Almost done. All right. So thank you for those inspiring words, Congressman Thompson. And thank you for your continued leadership and commitment to neuroscience. So now is when the fun starts, okay? That was fun too. Yeah, you're right. But now's when the science starts. So it's my pleasure to introduce Dr. John Nye, Director of the NIH Brain Initiative. Dr. Nye received his undergraduate degree in chemistry and biology from Pomona College and his PhD in biology from Caltech. He pursued postdoctoral research at Caltech and Columbia, later joining the faculty at Berkeley, where he spent over 25 years advancing neuroscience. As director, Dr. Nye leads the NIH Brain Initiative strategy and operations, driving scientific breakthroughs in brain science and applications to disorders like addiction. John.
Thanks so much, Mark. Also, thanks to Representatives Luttrell and Thompson and the entire Congressional Neuroscience Caucus for your support, and of course to Congress for supporting what we think is really game-changing research, and all of our sponsors here as well. And thank all of you for making the time to be here today. So the human brain, this thing between our ears, it's the most amazing organ in the body, the most complex, one of the most powerful, if not the most powerful computer that we know on the face of our planet. 86 billion with a B neurons or nerve cells that make hundreds of trillions of connections. Again, the most powerful computer, but it only consumes about 15 to 20 watts of power. So really an inspiration for all of us. Highly complex, but this complexity, this power also makes it so vulnerable to disease. And the issue that we have is we don't yet have the tools to understand how this magnificent machine works. And to put it quite simply, we cannot fix what we do not understand. And understanding the inner workings of the human brain, the secrets that it holds for us, is one of the scientific challenges of our lifetime. Now, as Congressman Thompson just alluded to, everybody here is touched by somebody with something going on with their nervous system. 1 in 3 Americans, over 100 million humans, is living with some neurological, neuropsychiatric, or behavioral disorder. So think about it, babies with epilepsy, young adults struggling with mental health crises and suicide, adults with Parkinson's disease, Alzheimer's disease, other degenerative disorders that are robbing them of their lives, and their families too, veterans with PTSD and suicide, and individuals with substance use disorder. Everybody here is being touched by this, if not themselves, a loved one, a close friend. We can't escape this. And cures cannot come soon enough for all of us. Now, millions of Americans are also living with chronic pain. Now, this is a neurological condition. It's not just something that's in your head, figuratively speaking. It literally has a biological basis. And again, if we can understand the biological basis driving these phenomena, we have a chance at being able to help folks that are living with this, which can really rob them of their life as well. Now, one can, in a simplistic way, think of the flip side of chronic pain as the dangers of addiction, right? When people need to use various medications to control their chronic pain, oftentimes using opioids. And we all know about the opioid crisis that's facing us. And again, we need to understand the biological basis of all this. Now, this involves a reward system, and my colleague, Dr. Nora Volkoff, is an expert in this. But the reward system is ancient. It's in pretty much all animals that we care to think about or study. And so we need to understand the basis, especially because behavioral disorders like addiction are often stigmatized. But it really comes down to biology, and this is how we really need to focus our attention. And again, we need to understand it so we can fix it. So where does the Brain Initiative come in? We are here to build tools to understand how the brain works in health, but then how to fix it in cases of disorders. So we are supporting the development of game-changing technologies and resources that are laying down the foundation through knowledge to develop these future cures and to bring them closer to us in our lifetime. And we're changing the way we're doing science. Given the complexity of how the brain works, one lab with one expertise cannot solve this problem. We need to we are bringing together biologists, chemists, physicists, engineers, behavioral scientists, clinical scientists. We're going to hear from these folks today, coming together with their different expertise to solve these really, really tough problems that could only be solved with this combined approach. And then very importantly, we're bringing cures to people today. We're going to hear about some really cool, really critical first-in-human trials that are paving the way for future therapies. So between this, providing these new resources and know-how, building more powerful teams with this great expertise, but also driving this toward cures, first-in-human trials today is what we're really about. And I think from the panel today, you're going to hear about some really amazing work, how this game-changing technologies are being applied and how the science will drive forward future cures. So today we have with us, let's see, starting out, I'll just state very briefly, we're going to hear about a really pivotal study, very important study being conducted at University of California, San Francisco. We have with us here Dr. Prasad Shivalkar, who's an associate professor of neurology, funded both by the Brain Initiative as well as the Helping End Addiction Long-Term Initiative, or the HEAL Initiative. This is a great example of a collaboration across NIH, breaking down silos to get these things done. Dr. Shivalkar was involved in a really important study looking at chronic pain, finding what we call activity biomarkers and using that information to help treat patients who otherwise had no hope of having their conditions alleviated. And with us today is one of the team's early research partners, human research partners, Mr. Edward Mowry, and we're going to hear about how this has changed his life. We also have with us Dr. Paul Kenny, who's the director of the Drug Discovery Institute, and is also now chair of neuroscience at the Mount Sinai School of Medicine. And he's using resources developed by the Brain Initiative to identify the cells and circuits in the brain responsible for driving addictive behaviors. Really game-changing work, and he'll tell us how he's going to translate these into clinical trials for humans. And then finally, here today with us is my good colleague, Dr. Nora Volkoff, director of the National Institute on Drug Abuse, one of the 10 NIH institutes and centers that's involved in the Brain Initiative. And she's a renowned researcher in the field of addiction and addictive behaviors, and also just an expert on the research that's being done in this area. So I'd just like to start off with a question to Dr. Volkoff, Nora. So from your perspective and decades of research funded by your institute, groundbreaking research that showed unequivocally that addiction is a disorder of the brain. It's a biological phenomenon that we have to understand. What do we know now that we didn't have 10 years ago? And how did the Brain Initiative help get us to where we are?
First of all, thanks very much for having me here. It's a great opportunity, and I love the Brain because it's a simple answering in your question is here we have an example about why funding, investing on basic knowledge, fundamental science, can actually transform the way that we address problems that we're dealing with. So in our country, we are facing the most devastating overdose crisis we've ever seen. People are dying, young people, from overdoses. And at the same time, we have not been able to provide sufficient therapeutics to help them. So how has the Brain helped us? There are two ways that it is already helping us, and there are ways that it's going to help us into the future. One of them is by providing us the knowledge about the diversity of cells and how those cells coexist and interact with others in different areas of the brain to form circuits and networks. So if I were to use a metaphor of how the brain works, it's the most complex networks of networks. And these networks are very, very dynamic, and they go from large clusters to smaller ones. And what we now know, for example, with respect to addiction, is that you are engaging circuits and systems that normally do not exist. So that drugs artificially have the capacity to sensitize certain systems. So by having exactly what the Brain Initiative has given us, a census of the diversity of cells in our brains, number one. And number two, a census of the connections. So how does a given neuron connect with what other neurons? That information is key and fundamental then to target interventions like what you're going to be seeing from Dr. Paul Kenny, to see if I manipulate specifically, exquisitely one cell type, I can modify the behavior, and that opens up the doors of therapeutics. So one of them is knowledge. And the other thing that is extraordinary that the brain has given us are tools and technologies. So what we are able to do right now, I'm a psychiatrist by training, so our treatments are either behavioral or medications, and that is changing, and it's going to be increasingly changing as we can now understand how the brain is organized, how it's functioned, and we have the tools in part developed through the Brain Initiative that allow us to tailor intervention to strengthen regions and circuits that may be affected by drugs or by other insults, so that we can buffer them and accelerate recovery. I think as we are speaking about human innovation, there's nothing that in and of itself cannot see us in the future utilizing these technologies, not just for treating addiction, researchers are doing that, but eventually actually coming to a cure for addiction. And I also see it too as a strategy to help us achieve recovery, to help someone whose brain has been damaged by drugs or insults actually strengthen it back so that it can optimally perform. So that's what I would say that the brain has given us. It has given us the knowledge and it has given us the tools.
Yeah, thanks. So we're going to get to the first part of your answer again with Dr. Kenny, who's really done some amazing work teasing apart these cells and circuits. And in terms of behavior and being able to cure addiction, but also in the present case, I'd like us to think about curing, at least really alleviating chronic pain. Dr. Civaco, tell us what drew you to the study of chronic pain. You're a practicing neurologist. You're one of the leading research centers in the country. Tell us how you got here, and how did this study evolve, and what brought you to working with Mr. Mowry?
Yeah, sure, John. Thanks, and thanks again, everyone, for being here and for the opportunity. Congressman Luttrell and Tom Thompson. I'm a neurologist. I work at the University of California in San Francisco. During my training, I got the impression, or I was actually very surprised to see how little attention there was actually paid to chronic pain, right? Pain, like actually Dr. Congressman Thompson said, it was a symptom. People recognized it as a symptom of other diseases. And it wasn't until, honestly, in the last decade that we started to appreciate that chronic pain is actually a disease itself. And when I say chronic pain, that we describe it as pain that persists for 3 months or longer. It's a little bit arbitrary, but the understanding is that there are fundamental changes that happen in the nervous system, in the spine and in the brain, that lead to perpetuation of pain. So in chronic pain, it's like the fire alarm is broken, right? And one, it's just in an on state, and we often can't find a fire, right? And so we don't know how to diffuse the alarm. So driven by this big gap in neurology training and in neurology expertise, as well as the lack of treatments, I really wanted to study it and see, okay, why are there not good therapies available? Number two, I was shocked to read that chronic pain, the number of Americans that develop chronic pain every year is more than the number of Americans that develop high blood pressure, diabetes, or depression, which is mind-boggling, right? The idea is that chronic pain is, you can't see it, it's often not visible. And two, doesn't lead to disability sometime in the future. It leads to disability right then and there. And people who have pain can't work. The relationships suffer. And so for me, I'll be honest, I'm a neurologist, but I'm also kind of a bleeding heart. And so part of my goal was to understand, all right, how can we treat the toughest of the toughest pain disorders? And so, the interest in research really led to me studying brain stimulation and working with partners like Ed here, funded by the Brain Initiative and the HEAL Initiative. And so there are a lot of young staffers here, right? So in science, it's the same. The key things for success is great mentorship, good scientists leading you the way, and two, teaching you how to responsibly use funds to discover basic science and translate that to really human trials to make a real impact.
So we're going to talk a bit about deep brain stimulation. So maybe you can help us unpack that. Deep brain stimulation has been around for decades, initially developed to treat the symptoms of Parkinson's disease by literally stimulating deep in the brain with electrodes, little zaps of electricity that can control these symptoms. And I remember just over 10 years ago when the Brain Initiative was first launched, there was this thought that it could be applied to more complex conditions like depression, OCD, PTSD, and chronic pain. And so tell us about that, because it's not just what you're doing is not, you're just not stimulating, but you're actually being able to read out what we call biomarkers, activity biomarkers. So if you think about the field of cancer research, why has there been such progress there? Because they have biomarkers. Clinicians now have, because of basic research, clinicians can now identify when a patient has a cancer and what kind of cancer it is and tailor a treatment for that. And what's amazing to me, 10 years ago, I remember people saying, we had this idea, the group that you work with, they had this idea that they could develop these biomarkers to develop stimulation therapies that would help in these more complex neuropsychiatric disorders. And I thought you all were crazy. But tell me how you went from crazy to reality, because we're going to hear about reality today.
Yeah, absolutely. You're absolutely right, John. Pain is in the brain. We know that it involves brain circuits, right? And so deep brain stimulation, like you said, which is an FDA-approved therapy to treat disorders like Parkinson's disease and movement disorders, and even epilepsy, is appealing, right? Because you have a direct line to the brain where you can actually interfere or adjust the language of the brain. And the real-time language of the brain, in many respects is electricity, right? And so we have electrical signals that are very complicated, that we're just beginning to scratch the surface of, like Nora said, networks of networks. How do you model such things? And so in Parkinson's and in epilepsy, often in Parkinson's at least, there seems to be more homogeneity, right? There seems to be a single target that works for many patients, or two targets that works for many patients. Pain, unfortunately, is much harder, and there's no pain center in the brain, and so pain is quite distributed across the cortex, across older parts of the brain, like the thalamus and parts of the basal ganglia. But really, what we aimed to do is, I don't know if that's a fire alarm. Yeah, exactly. Perfect.
Stimulation.
Yeah, that's good. Yeah, I recognize that. So what we aim to do is, all right, in patients who are suffering from chronic pain, we not only, like you said, John, not only wanted to understand where can we treat, but where can we read out? Can we understand by recording from these signals in the brain? Can we identify a biomarker? And this was really only possible because, one, private industry was developing devices for the first time 10 years ago that allowed us to record, to actually use like a microphone in the brain as opposed to a loudspeaker, just delivering electricity. And so we worked with Medtronic, right? And that partnership was really only made possible by the Brain Initiative. And so they made devices, and this is pretty amazing, that they had an entire research arm within a company. It was high risk, high reward research arm that developed because of the Brain Initiative, because the Brain Initiative allowed folks who are studying Parkinson's and depression and OCD to then work with Medtronic, where then they had the incentive to develop this research arm. That device 10 years ago that allowed us to perform the preliminary biomarker studies where we could sense signals in people's brain and record biomarkers of pain, led to a second device. And it was never commercial, right? So these are in-house research grade devices that we had private contracts with Medtronic for. And that second generation research device is actually been a huge success, as evidenced by Ed sitting right here next to me. And believe it or not, that second generation research device actually turned into a commercial device that got FDA approval for closed loop or real-time adaptive brain stimulation just a few months ago. And so that was a 10-year journey, and we were so lucky to be on the forefront of it because of.
Yeah, 10 years in this game is pretty quick. So tell us just real quickly how Mr. Mowry, Ed, came to you. And Ed, we want to hear your story to how this changed your life.
So yeah, it's a good question. One thing that the HEAL initiative allowed us to do was to recruit patients from not just California, right? Because it's more financially feasible, right? If you have local patients, you don't need to fly out. But thanks to the HEAL initiative, we were able to recruit from all over the country. And actually, we had 2 patients from Texas, another patient from Florida, and another patient from Michigan. And Ed, came to us from Albuquerque. He reached out pretty early, right? And what we were looking for, deep brain stimulation is invasive, right? We have to drill holes in the skull and implant electrodes like they do for Parkinson's. But we actually designed a study where we performed an exploratory period. We took a page out of the book for epilepsy, right? So if folks have epilepsy and you don't know where the seizure's coming from, you can insert multiple wires in the brain and try to map out, okay, where is the focus of this seizure? So we used a similar playbook for chronic pain, keeping or inviting patients to essentially sign up for the study and stay in the hospital for 10 days, where we systematically mapped out with stimulation and recording what might work for them and what might not work to provide significant pain relief. All these patients, or really these partners, had been failed by all available therapies. Many of them have had chronic pain from stroke. You can have a stroke that fills in burning pain in half your body. Some patients had spinal cord injury. Other patients had a rare syndrome known as complex regional pain syndrome. And so, they had to have been failed by at least 3 medication classes, but in real life, they failed more like 20 medications. Injections didn't help. Spinal cord stimulators didn't help. So even the cutting edge therapy that was out there didn't help. And so there was an extensive screening process where we made sure people understood the risk, were willing to be pioneers. And Ed really, was one of the pioneers.
Ed, tell us about your story as one of the pioneers here.
Hi, thanks for having me. Edward Mowry. I am founder and guitar player and vocalist for a metal band called Tiwanaku. It's Tiwanaku. I started my pain journey when I was in high school. I was a soccer player, downhill skier. I did a lot of rock climbing and rock falling. And eventually it got to where it was pain all the time. And I'll be having my 35th surgery for my left knee to be replaced in July. I've had C3 to T1, L4 to S1 in my spine, all fused, disc replaced, have had my knee replaced twice. The 3 brain surgeries I had to have to get the deep brain stimulator. I was basically 5 years ago, I was sitting in a wheelchair and in and out of a walker and crutches. And once they finally did all the orthopedic fixes to my body, I was still in pain. They replaced my knee, and a week later, I was in the most excruciating pain I've ever been in my life. And I went years, on and off of opiates, on Lyrica, gabapentin. I was on 180 milligrams of morphine at one time. And now I am down to almost 30, and that's as needed. Thanks to the brain stimulators and Prasad and UCSF. When we were in the hospital for the 10 days, it took until the last couple of hours for them to actually find the points in my head that would kill the pain. And all of a sudden, we've got a video of me going into euphoria because every bit of pain stopped in me. I went down to a 0, and I hadn't been in a 0 in years and years and years. And eventually, after we did the study, I came back. They put the deep brain stimulators in my head permanently, and then I came back a month later, and they turned them on, and I left the study that day, and got to my hotel room, and I woke up 8 hours later in the middle of the night, looking out my window, going, "What happened?" And it almost felt like all the muscles on my bones were falling off, like ribs do fall off the bone. But then once I got used to it, it's been amazing. It's 100% changed my life. I mean, at first I waited a year and a half to make this happen with these guys. There was no way they were going to do brain surgery on me. And it got so bad. Like I said, I couldn't barely walk. Some days I couldn't even get out of bed for the whole day. And, you know, pain, just think about the worst pain you've ever been through and then multiply it by about 200. It's the worst pain you could possibly think of. And the thing that took me a long time to do was to wrap my brain around the fact that pain comes from the brain. And this was manufactured pain because I'd been in chronic pain for so long orthopedically. They finally fixed me, and my brain said, whoa, we need to add that pain back in for you. And it was the worst I've ever been through. And they figured out how to kill it. I'm in zero pain sitting here right now.
You know, it's hard for me to imagine to be in that state for so many years, but I'm trying to wrap my head around what it was like just all of a sudden it being gone.
Oh, it was like the best drug I've ever taken.
But it wasn't a drug.
It wasn't a drug. And that was the
Wasn't a
great thing
drug.
about it. You know, and I mean, I was looking up at the ceiling just going, oh my God, you know, it's gone. It had been with me for, you know, years and years and decades.
But you were motivated. I mean, you know, I'm thinking about, for me, for my loved ones, the thought of having, quote unquote, brain surgery is pretty scary, right? I mean,
It
not afraid to
scared me
admit
bad.
that, but
I was
you were
really scared.
motivated.
Yeah, what motivated me was 24/7, 365 pain. Even when I was asleep, I would wake up in the middle of the night screaming in pain. And, you know, it's just, you know, that doesn't happen anymore. I've 100% gotten my life back. I'm about to get off disability. I called my disability lawyer and asked her, how do I get off disability? And the phone went silent. And I'm like, are you there? She's like, oh yeah, I'm here. She's like, in 25 years, no one's ever called and asked me that. And yeah, that was really, really impacting it for her and for me too.
What also impresses me about this, and with anybody that places trust in another human, their life, that's something. But this is kind of new. This is bleeding edge stuff. Nobody had ever tried this before, and you must have had either an amazing amount of desperation, which you might have, but also an amazing amount of trust in the team at UCSF to work with you on this.
Yeah, absolutely. You know, for the longest time, you know, I've been treated like a drug seeker. I've been treated, you know, like I was addicted. I've never been addicted to these medications. It's always been there just for pain. And now, like I said, I was 180 milligrams of morphine a day, 450 milligrams of Lyrica a day, and Cymbalta, and I'm almost done with the morphine, a third done with the Lyrica, and the Cymbalta's next.
Yeah.
I'm going to get off all the medications. That's my motivation, was to get off every medication I'm on. I was on 17 medications at one time. I'm on 4.
It's truly, like I said, you know, when folks were proposing that we could do this 10 years ago, I was pretty skeptical. And to be sitting here with living proof that it works, there's enough trust by you of the process to make it work is just amazing. And I want to get back to something that Dr. Scirocco said. I mean, basically, the game here was to find what part of the brain is responsible for this. And it wasn't obvious, unlike, say, in Parkinson's, the motor symptoms of Parkinson's. We know where things are going wrong. And what's amazing is that once you've been able to find that locus that's responsible for the pain and be able to read out these electrical signals, by the way, with really sophisticated computer algorithms. We kind of glossed over that. That's amazing, but it's still, you know, putting electricity into parts of the brain, it's on the one hand very sophisticated, but on the other hand, it's a pretty blunt instrument. And so Dr. Kenny here has been using some really amazing molecular tools to discover these a master regulator of opioid reward and addiction. And Could you tell us a bit about that and where that research is going and what got you there?
Yeah, absolutely. I was struck by Ed's comment that when the pain was turned off, it was like the best drug he'd ever experienced. So opioids, of course, we've known it. Opioids have been used by humans for hundreds, if not thousands of years. And the reason being they're remarkably powerful. If you're in pain, opioids will absolutely turn that pain off. Very, very good at doing that. But the problem is they very quickly lose the power to accomplish that because you develop tolerance to the opioids. It's a major problem. The other problem, of course, is that when you use opioids, generally, they kind of make you feel good, they're pleasurable, and they have addiction liability. And as you use them for their rewarding properties, you can develop dependence. And honestly, we know very, very little about how all those processes occur. We do know that drug dependence, substance use disorders, they're brain disorders, in no small part from the work from Dr. Volko. If you look at the brains of someone who's been using opioids extensively, they're fundamentally different. They're completely different in their operation. They're completely different in terms of how the neurons work. The gene expression profiles are completely different. So one question my lab asked is, how do opioids work? Simple question, you would think. And at this stage in the field, you would think we'd know. But the brain is, as John said, our brain's remarkably complex. It's got billions upon billions of cells. You can break them down into groups of cells, but even then, those groups, they can be fundamentally different. And unlike the heart, unlike cancer cells even, we know remarkably little about the brain is wired, about the cells that are contained in the brain. So perhaps it's not surprising that we don't really understand deeply how disorders like drug addiction come about. So like I said, we asked, which cells may actually be involved in responding at the very earliest stages when you experience an opioid? How do opioids get into the brain, act such that they elicit pleasure and may drive the development of addiction? So we use various tools that were developed by the Brain Initiative to be able to basically say, if we inject a mouse with an opioid at a dose that we think is rewarding, I'm going to put aside all preconceived notions about where I think they're acting based on the older literature. Let's just ask, by basically examining every cell in the brain and see which ones show a change in activity when an animal is presumably experiencing the pleasure of an opioid for the very first time, identify those cells that show changes in activity, then see what's inside them that can explain that change in activity, and then go in and manipulate those cells to see if we can modify the animal's response to opioids. And anyhow, long story short, what we found using these tools that have been developed by the Brain Initiative, we found that, to my surprise, that one of the most complex areas of the brain, the prefrontal cortex, actually contained a population of cells that express mu opioid receptors, which, when you activate mu receptors, that can be immensely pleasurable. What we didn't expect was that those cells would be in the prefrontal cortex that would be involved in regulating acute behavioral responses to opioids, because this is an evolutionary, very advanced area of the brain, yet responses to opioids is conserved all the way back through species development. So that was surprising. Another thing that was surprising is that the cells that express these opioid receptors are actually really advanced glutamatergic cells that project to other brain regions. And why that was surprising is any opioid receptors that are expressed by cells in the cortex are thought to be expressed by these local cells, these GABA neurons, not these long-range projection neurons that send information elsewhere in the brain. And then, anyhow, we ended up taking the receptors out from these cells in this part of the brain to see what would happen. And not only did we block the rewarding or pleasurable euphorogenic effects of opioids, we actually reversed the hedonic valence of opioids. What does that mean? Opioids were no longer rewarding, they were actually aversive. Animals didn't like them anymore. So we think that these cells play a key role in basically permitting opioids to be rewarding. And why that's important is we've sequenced these cells using the tools that John had mentioned, so we know what they express, which means that we are now in a position where we may be able to design entirely new classes of therapeutics to be able to manipulate the activity of those cells and ideally be able to block the progression of substance use disorders. Maybe in people who have a substance use disorder, modify the activity of these cells and reverse the pleasurable effects, stop the motivation that drives people from using drugs of abuse. And so we've actually identified a whole range of receptors other than the opioid receptor that are in these cells that we're actively trying to develop new molecules for, new drugs to help treat drug addiction.
So one question we always get, and frankly, one question we ask ourselves, I mean, this is beautiful science. It's absolutely stunning and gorgeous. But we always ask ourselves, how's this going to help people? What is the bridge between this, beautifully laid out mouse model to helping people with these disorders?
Yeah, that's foremost in my mind. And the reason is that we just don't have the therapeutics available to help people with substance use disorders. They are brain disorders. We should be able to treat these disorders like you can any other brain or any other peripheral disease-related disorder. But with support from Dr. Volko and NIDA, we and others are actually trying to take that next step, where if you identify what you think is compelling evidence implicating a protein in the brain, a gene in the brain, or a cell type in the brain, can we manipulate that target and develop new therapeutics? Because honestly, if we don't do it, it's probably not going to happen. Without the support of the NIH, NIDA, and the Brain Initiative, there aren't large pharma companies who are developing therapeutics, because if they were, we'd already have them. We have a huge knowledge base to guide us in terms of what are probably good targets for the development of new substance use disorder therapeutics. So it's people like me who try and identify or understand how addiction is coming about, who are trying to take that next step and develop therapeutics. And we have a range, a pipeline, if you will, of hopefully new drugs being developed in my lab that we're trying to move into human clinical trials and translate the basic knowledge into new treatments that can help people.
I'm going to give you a perfect example about how this is so very valuable and the thinking that goes behind it that we're now using for the treatment of addiction. It was approximately, I think, close to 2 decades ago, maybe 15 years ago, where there was a study that was published in Science where they showed that people that have suffered a stroke into the insula completely forgot to smoke. And so this was touted as here we have come for a cure of smoking. And so researchers have been trying to understand, well, first of all, what is the role of the insula? But also the other question that clinicians started to see that not only lesions in the insula produce these cures from addiction, but in some people, those strokes or traumas in the insula didn't cure the addiction. And what researchers now can do, because of the mapping of the connections, is in a given person, identify how that particular area where there's damage connects with other areas of the brain. And as a result of that research, it's not possible to identify regions exactly like the ventral prefrontal area here. And when Paul is saying, well, this is very important for reward, why is it? This area of the brain provides saliency value. So the same stimuli can be rewarding or it can be aversive. If I'm very, very hungry, food can be extraordinarily rewarding. If I'm completely satiated, it can be aversive. So that system actually is profoundly affected in addiction. And what we now know, for example, it is damage, and that interventions that can strengthen it to enhance the value of other reinforcers actually appear to have promising as therapeutic. So this is an example about how basic knowledge on cells, connections, animal models, and ultimately tools that are developed, like the one that has been implanted to treat pain, can now start to give us a completely different possibility for treating brain disorders in general, whether it's Parkinson, as has been said, or whether it is depression or addiction. And this is just the beginning.
Yeah, it is the beginning. What's fascinating to me is Dr. Kenny, your group identified one of these many thousands of cell types in the mouse brain and its connections. To put that in perspective, the studies that we supported in Brain created what we called the Whole Brain Cell Atlas. And in the mouse, we identified about 5,000 cell types. So it wasn't just a matter of a needle in a haystack. It was a matter of having the tools to identify that one type. And but probably represented more than 1 in 5,000. But nevertheless, you get the idea about the odds that are against us for trying to understand the complexity and then to understand where it projects to, which is critical because the brain is a computer. Computer is made up of components that are wired together. And if you understand how they're wired together and how the information goes through it, then you can kind of figure out how it works. And most importantly, how to fix it when it's working in a way that you don't want it to. And just to give you another idea of the complexity, recently was just published a series of papers from what we call the Microns Project, Machine Intelligence from Cortical Networks, that characterize 1 cubic millimeter of a mouse cortex. So 1 cubic millimeter is about the size of a grain of rice. A mouse brain is 500 times that size. A human brain is another 1,000 times that size. And in that 1 cubic millimeter was 500 million connections characterized, 4 kilometers, 2.5 miles of axons connections. And we, in the BRAIN Initiative, BRAIN stands for Brain Research Through Advancing Innovative Neurotechnologies. We think big, and our goal is to map out all the connections in a whole brain so that we can better use this as a resource. So the next time you discover a cell that you think might be involved in addiction or chronic pain or whatever, you can go to that resource and push a button and find it and be able to be on your way and then test it and then translate it into human use. The other piece of this is animal models, I think justifiably, have been getting some criticism because a discovery made in, say, a mouse doesn't always translate into human. And the knock on us is it's not good enough to cure a mouse, you have to be able to cure a human. And what we found through our cell atlasing programs is we actually now can tell if we find this cell type in a mouse, can we find its counterpart in a human? So I think the day will come where the first thing people will ask is that cell, is that receptor, is that protein? I'm seeing it here in this really interesting behavior. If I don't find it in a human, that's when I'm going to stop and pivot to find something else. And in fact, many drug trials have been shown to fail because they got the wrong target, a target that didn't exist. So, I'd love to hear more about how we can translate that out to help people. We've had some conversations beforehand. I think everybody's heard about these current miracle drugs, Ozempic, these GLP-1 receptor drugs that, in addition to helping people with weight loss, diabetes, blood pressure, almost everything, but they also seem to be involved in addictive behaviors. And why don't I let you two talk about where we might go with that? Because if we understand where those receptors are, we might be able to better control some of these errant behaviors.
Gentlemen, I can jump in by sort of how did this come about. For more than a decade, we have been funding researchers like Dr. Paul Kenny, who are looking at animal models of addiction. And they had shown that the first generation of these drugs were able to actually not just decrease the amount of drugs that these animals were administering it, but also decrease the likelihood that they would relapse. As more and more people were given these medications, there started to be reports of people saying, well, I'm on this very strange, I no longer have desires to drink, or I no longer have desires to smoke or to take drugs. And that actually has opened up all of the doors to try to understand whether, in fact, these medications hold potential for the treatment of addictions. And the data from randomized clinical trials, for example, in alcohol, show that it does significantly reduce heavy alcohol drinking days. There is data, randomized clinical trial, to show that also it appears to be reducing tobacco. And we're currently doing the studies for opioid use disorder and cocaine use disorder. But to me, the aspect that I do think that I would like to highlight, because the brain has allowed us to start to look at it, we tend to look at our brain like a separate entity, but the brain is highly communicated with all of our body. And imagine we pay attention to visual system, to auditory, but trust me, all of the information that comes from your brain, the chemicals, your heart rate, your lungs, are massively important for how the brain works. And understanding how these systems, peripheral systems, ultimately influence the human brain actually tells us, in a way, it's not surprising that when we have a medication that profoundly affects metabolism in all of our body, inflammation in all of our body, we're seeing benefits in addiction, we're seeing benefits in dementia, we're seeing benefits in pain. There are actually reports to also potential interest for antidepressants. But ultimately, at the end of the day, understanding that how that comes about is crucial. And that's where actually Paul has done really some marvelous, elegant work.
Yeah, Nora made a really good point, a really good analogy, and that is about saliency. So, food, if you're hungry, can be remarkably rewarding. If you're full, it can actually be aversive. And then I think you could extend that analogy into drug addiction, actually, because quite often we think of addiction as being this desire to obtain the pleasurable qualities of a drug, and ultimately it's a disorder of, reward seeking. But when you use a drug, it quite often there's patterns of drug use, and then people are, for smoking, for example, they don't smoke till all the cigarettes are gone. They control their consumption. And the reason I tell you that is because the flip side of the coin, in terms of saliency, where there's an aversive component or there's a satiety component, there's an appetite component, is probably real. That you use drugs because, yes, they're pleasurable, but you control your consumption because you don't want to go too far. We know a lot about what drives the motivation, the reward-related systems. We know very little, even less, relative to reward systems in terms of where the notion of satiety comes from in the context of drug addiction. It was with that notion of satiety that many labs, mine included about 10 or 15 years ago, started looking at satiety modulators. And we hear a lot about Ozempic now in the news. Ozempic is a GLP-1 receptor agonist. We've known about that system for 20 or more years. We've known that GLP-1 receptor agonists, when you activate these receptors, will actually promote satiety, will decrease appetite. We asked, could you decrease the appetite for a drug of abuse? And you absolutely can in an animal. If an animal is consuming alcohol, cocaine, nicotine, opioids, you treat them with drugs like Ozempic and other GLP-1 receptor agonists, the appetite for the drug is quelled, it's decreased. So we predicted a long time ago, many labs who are working in this space, that perhaps we'll see a decrease in the appetite for drugs in people when they're using these drugs, GLP-1 receptor agonists. Lo and behold, a next generation GLP-1 agonist like Ozempic comes along, and what we're seeing now in humans is what's being predicted from the work that's being supported by NIDA, that you would quell appetite for addictive drugs. So it's not at all surprising from the preclinical literature that we're seeing benefits of these drugs. Now the challenge is, how do they work? Which systems in the brain, which cells in the brain are responsible for quelling drug appetite? Can we understand these really poorly understood circuits? And if we can, then we might be able to develop entirely new classes of therapeutics that tap into these systems, maybe even better than Ozempic does. So using the tools from the Brain Initiative, that's something my lab has spent the past 2 or 3 years on. Where exactly are the GLP-1 receptors in the body and in the brain that explain this quelling of drug appetite? Can we identify the cells that express those receptors, understand what else they may express, and really try and kind of either make Ozempic better, so it really does work in people who have substance use disorders, or get next generation Ozempic that can target these cells, these circuits, and have real impact in people suffering from substance use disorders.
Yeah, it's amazing. I often refer to Ozempic and its similar drugs as miracle drugs, and they are a miracle in terms of what they're doing, but it's not magic. There's a biological basis for this, and if we understand how it's working, we can hopefully get better drugs to tune these specific conditions without other unwanted side effects. So that's crucial, I think, to be able to get these out and to make them accessible to people who need it at an affordable price. But this brings me to something that's always bothering us in these tools we're developing. How can we actually get them to help people today? In the case of this deep brain stimulation, as we see in this first-in-human trial done at UCSF with very highly valued partners like Mr. Mowry. Prasad, this first study that you all did is about half a dozen patients or so. So this is great. We changed their lives, and I think the majority of those patients, very successful. I think it's fair to say we've changed your life, and you're in a different place. But tell me about, the challenges that the two of you, you've been a team in this, truly a team. And how do you see, what is it going to take to reduce the barrier from the clinician side? What science do we need to promote to make this accessible, not to half a dozen or to 20 or 100, but to thousands of the millions of people who need it? And, what was the barrier for you to get into this? You had to go through this huge, I think, mental challenge to do this, putting yourselves in other people's shoes to the extent you can. What is it going to take for people to not have to agonize over, oh my gosh, do I need brain surgery, to get to something different? I'd love to hear, you two have worked together, I'd love to hear your perspectives on this.
Make it wireless.
Yeah.
That's pretty much it. It was the brain surgery was the barrier for me. I mean, like I said, it took me a year and a half, and then finally I gave up and gave in and went through with it. And I wish I would have done it a year and a half beforehand. I'd be that much further in life.
Right. But you've been a pioneer, and maybe you can tell us about thinking about how you can use the information, the data, the know-how you developed to create minimally invasive or hopefully non-invasive treatments.
Right. Yeah, absolutely. I mean, I think making it wireless is key, right? But sometimes, often actually, you will spend much more money to explore and to actually understand the phenomena and to describe pain circuits and even an approach that is a proof of concept that this works, this is feasible. In a similar vein, sending very sophisticated satellites, or not satellites, sending very sophisticated rocket ships with robots that are, highly expensive to Mars is required before you can send any humans, right? And it might actually be cheaper in some ways to send humans. And what I'm saying is the development of the technology, the establishment of the risks, characterizing the landscape is always going to be more expensive. And so I think that what the BRAIN Initiative has invested so far is amazing, right? We're doing this high risk, high reward study. We have amazingly and surprisingly proven, I think, that this is feasible. It's feasible to, within patients, target personalized circuits, right? People are different, right? And especially how people suffer, how people experience joy. And so I think we've demonstrated that we can identify signals in individual people that help to track their personalized pain, right? And other attributes of their pain. So how do we make this wireless? One, developing hardware, right? So we were lucky enough to work with Medtronic and have this partnership with a company that's been well established and kind of a powerhouse. Since 10 years ago, the neurotech space has exploded, right? I mean, I can't even count on two hands how many new invasive neurotechnology companies, startups there are, right? There are new companies to do recording, stimulation, decoding of speech, decoding of thought. I mean, there's, to play video games. None of it's commercial yet, right? But the idea is the BRAIN Initiative has inspired, I think, a big industry already focused on invasive treatments. Following that wave, there are already a few startups, that I can name that are doing new non-invasive types of brain stimulation that we never even dreamed of 5 years ago. And for example, something called temporal interference stimulation, really amazing but simple concept, right? The idea that you can provide radio frequency stimulation, on one side of the brain, radio frequency on the other side of the brain, and direct the individual radio frequency fields such that where they interact, right, and the frequency offsets by which they interact will selectively stimulate just that part of the brain. It seems like science fiction, and I'm pretty sure it's described in Star Wars, Star Trek. Which is, it's funny, a lot of inspiration comes from how to put this. Life often imitates art, right? And Star Trek is very prescient. But what's even more notable, I think, about such technologies, temporal interference stimulation, and now even low intensity focused ultrasound, right? Is that things that were sci-fi or just a dream decades ago have now, in animals, been demonstrated to have some real biological basis for modulating circuits without even making an incision in the scalp. And so I would say, right now we're at the stage where yes, there's startup companies developing to hopefully develop these tools to be able to create real therapies without brain surgery. But the real deep work in animals isn't done, right? Because we still have difficulty using these non-invasive tools in many cases to treat diseases that we can treat with invasive tools like Parkinson's and epilepsy, right? So we need to understand, we know these technologies can penetrate the skull and penetrate the scalp. We need to understand fundamentally what are they doing at the cellular level, at the circuit level. And once we have, I think, a better grasp of that from a basic biology perspective, it's not a matter of, if we can figure this out, it's a matter of when. When are we going to be able to just to say, okay, we now understand what this is doing. Let's apply it.
Yeah, and these are great points, and we are actually starting to see some success with these so-called non-invasive techniques using focus ultrasound, using magnetic stimulation, transcranial magnetic stimulation, and the reason why, in cases where it's beginning to work, is because using the more invasive studies and imaging studies, we've actually been able to identify where you want to be aiming the stimulation. So it's really, really critical. I think this is going to be the key. With the early pioneers, with the invasive studies, those who are trusting and courageous enough to go through these with us and to help teach us, I think this will be the basis for these future endeavors. So I think we're coming on time. We wanted to leave some time for questions from the audience, if we may. So Katie, should we open that up? Mark, you want to moderate that?
Can I ask a question to Ed?
Yeah, sure, of course. You've been waiting, sorry.
Yeah, no, I mean, because the story is so very unique. And I also got captivated by your statement that actually when the pain was gone, this was the extraordinary sense of well-being. And I think that it tells us at the essence of really ultimately what pleasure and reward is, how they are context dependent. So, but that was when the pain was gone. So since then, there's been several years. And how do you react? Do you still have this sense of well-being? What do you do to actually promote it?
I still have really good sense of well-being. And ever since back then, my attitude has always been 100% positive and 100% trusting in these guys. It's going through that. I still have that euphoria, but it's I'm sort of used to it now. I can step on a Lego and I still have pain. I still get pain in my feet from the CRPS and from the neuropathy, but the pain stimulator turns on and turns it off. I feel the pain come and I'm like, oh no, boom, it's gone. So that's taken a little getting used to, but I can handle it. It's compared to what I was dealing with before. It's night and day.
Thanks.
Yeah, it's amazing. And I have to say it is, I don't mean to sound cheesy, but it's inspiring to us to keep going on this.